So I will try to start my class today. I will take up a new topic, that is differential gene expression. This is possibly the last part of my syllabus. And any other problems, you people are free to contact me for your queries and questions. But today may not be the last class because this is a long chapter. It will take 2-3 more classes, but I hope by the end of January I should be able to finish, or by beginning of February this should be finished. So I think from now till the upgrade is problem has been solved, we are going to use Google Meet. And as usual, for me it hardly makes a difference. I will send you the recordings as I do. And so I'm just hold on. I'm just recording all your attendance, the people who are right now here. Atika, Abir, Orpita, Oyon, Jahanara, Mollika, Mohor, Papia, Partho, Ria, Shalu, Sharoda, Shattoki, Shoma, Shreya, Shubham, Shushmita, Shopnil. So these are the people right now in I will again at the end take the attendance once more. It will be recorded and I will put my values accordingly. So there are right now 18 students who have attended in your total class at the beginning of the lecture. So let us with this let me start presenting my screen. I never face internet problems, so today I am facing tremendous internet problems. So bear with me. I am using a Geo connection right now from my mobile, and this is one of the most unreliable connections present. I'll just start my chapter. This is Gilbert. I'll be teaching from Gilbert. I'll be teaching from Gilbert. Gilbert is a good book. Just hold on. Let's see, somebody is entering the class. Shubham Koviraj enters. Okay. So let's get back to the class. Okay. Specifying identity. We had finished pattern formation. So now we are going to study differential gene expression. That is chapter 3 over here. So the word differential, it means different. The word gene, you know, gene and its expression. So in all the cells of an organism, you find that in all the cells, it is assumed that all of them have the same DNA. So your eye cell has the same DNA as your ear cells, as your skin cells, as your hair cells, as your kidney cells, liver cells. They are supposed to have all the same DNA. This is assumed because it all started from mitosis of a single cell, that is the zygote. The zygote started to do mitosis, and from one cell, fertilized one zygote, you have several such cells present. Now the important this is the concept of genomic equivalence, so that each cell has got the same set of genomes in your body. Now this poses a very interesting question. If each cell has got the same set of genes present, then the question says that why is a heart cell so different from a muscle cell, and which is so different from your skin cell, which is so different from your eye cell? So why is it so different, or why are they different? Why are the functions different? How does this difference come? So this is what you study in differential gene expression. So the answer to why it is different is because the set of genes may be the same in all the cells, but all the cells are not expressed similarly. Your muscle cells express set of proteins specific for muscles. Although it has got the genes present for your nerve cells, but still, it's the muscle cell proteins which are expressed. Same with that of the nerve cell. It will not. It has got all the genes for the muscle cells, but it will not express the genes for the muscle cells. It will express genes for only the nerve cells. So this is a differential exercise. You study in this chapter why and how does this sort of regulation happen. In this part you can overlap this particular chapter with your molecular biology paper, gene expression, eukaryotic gene expression. This chapter has got a lot of overlapping topics in this. So this is what, so the concept of genomic equivalence says that somatic cells in the nucleus has the same number of chromosomes. That is, your nerve cells has the same number of chromosomes as that of your liver cells, as other somatic cell nuclei. And so the answer why cells are different is differential gene expression. So differential gene expression, it can be defined as the process by which cells become different from one another based upon the unique combination of genes that are active or expressed. So there is a combination of genes that are active or expressed in a particular cell at a particular time also, at a particular stress also. Under heat stress, only heat stress proteins are expressed. So this is a differential gene expression. In seeds you will find a set of proteins expressed. In leaves you will have another set of proteins expressed. So this is differential gene expression. So every somatic cell of an organism contains the complete genome established in the fertilized egg. In molecular terms, the DNA of all differentiated cells are identical. The unused genes in differentiated cells are neither destroyed nor mutated. They retain the potential for being expressed. In animals, this is a very difficult to imagine because of the power of regeneration and totipotency. They reside in stem cells, because your nerve cells cannot de-differentiate and become a muscle cells, although it has got everything. But still, in your certain stem cells or in plant cells, you can make a plant from a leaf cell. You can de-differentiate it and you can re-differentiate it back into another plant in tissue culture. So they retain the potential, that is, the genes are not lost. In of course in matured B cells and T cells there are certain losses which take place, but in most of the other cells it is not lost, it is present, it retains the potential. Only a small percentage of the genome is expressed in each cell, and a portion of the RNA synthesized in each cell is specific for that cell type. So there are two types of genes which are expressed. One are the housekeeping genes. They are expressed constitutively at all times in possibly all of the cells. And then there are the specific tissue specific cells or factor induction specific cells. All these cells and genes which are present, they are different. So only a very small percentage of these all the total genes in a cell is expressed, not all of them. Only the nerve proteins are expressed in a nerve cell, not the muscle proteins. You have the complete complement of all the genes of the body present in one cell, so that is not expressed. So by the late 1980s, it was established that gene expression, it can be regulated at 4 levels. First is the differential gene transcription. At the level of transcription, some are transcribed, some are not transcribed. Some genes are transcribed, some genes are not transcribed. So this is how it goes. Next is selective nuclear RNA processing. And this nuclear RNA, these are processed, these are not processed. This regulates which of the transcribed RNA or which part of nuclear RNA are able to enter into the cytoplasm and become messenger RNA. So there is a specific, last day I explained something about splicing. So there is a type of splicing that particular part of the transcript, it is spliced out, so that doesn't go to the cytoplasm to be translated into protein. So controlling the regulation of splicing, for example, in your in several cells, there is the in sex determination in Drosophila, there is a term known as alternate splicing which takes place. You have, say for example, two introns. So two introns means they are you must have the flanking region of the introns are the exons. So exon 1 intron, exon 2 intron, exon 3. So basically there are three exons and two introns in between. So the two introns are spliced out and you have only exon 1, exon 2, exon 3 attached with each other. So this can very well be the case that the intron 2 is not spliced out, only the intron 1 is spliced out. So the final transcript becomes exon 1, intron 1 is spliced out, exon 2, then you have the intron 2 present, it is not spliced out, and you have the exon 3. So you can have these two types of alternate transcripts present. This will result in a different type of protein being present, because the intron then when it gets translated, it will give you few different type of amino acids. So different cells, for example your LDH in muscles and LDH in liver, they are different, they are totally different. And this is a very important aspect, because both of them are LDH, but muscles and liver, they are different. They have got different because they are alternatively spliced. Some part of that particular LDH is not present in muscle, which is present in say for example liver. The glucokinase hexokinase in glycolysis, that's also another example of certain some different types of isotypes which are present. So there is a selective nuclear processing, because of this there is a type of regulation, a different type of gene is expressed. Then the third is selective messenger RNA translation, which regulates which of the mRNA in the cytoplasm are translated into proteins. So all for example your ribosomal messenger RNA which is formed, the ribosomal RNA which is formed, that is not translated. There are several such non-protein coding mRNAs which are transcribed, but they are not translated. So there is a selection. This is going to be translated, this is not going to be translated. So there is, at the level of translation, there is a sort of regulation which takes place. Then you have selective messenger RNA translation, which regulates which I told. Then the fourth part is differential protein modification. So some proteins are methylated, some proteins are ubiquitinated, some proteins go through other levels of modification, there are different types of modification. That also makes a protein phosphorylated protein is active and non-phosphorylated protein is not active. So there are 4 steps of regulation which takes place. And because of this type of regulation, even though the same number of same gene sets are there, some are expressed, some are not, because this type of regulation is different in different cells, and therefore you have a differential gene expression. Some genes, such as those protein for the globin protein subunits of hemoglobin, are regulated at all the 4 subunits. So the next day I will talk about all these, and mind you, this is also going to be a class for gene expression also, eukaryotic gene expression. That is what I am teaching. It's the same. It will be an overlapping part. Okay, let us go. Let's see, somebody is coming in, I believe. Nazia has entered. So let's take a intermediate attendance. Atika, Avir, Orpita, Oyon, Jahanara, Mollika, Mohor, Nazia, Papia, Paramita, Partho, Ri, I think this is Shayantoni Das, Ria Kormokar, Shalu, Sharoda, Shattoki, Shoma, Shreya, Shubham Shah, Shushmita Ruidas. So these 23 people have attended this class. So let's get back. I just don't want any of you to miss out because of this. But unfortunately, by rules, this is your official attendance. Whether you have a network problem or not, I believe there is no excuse. You have to ensure there is no network problem. You can be late because of network problem, but at least your name should come up. As for this, you will get the recording. All the lectures, you will get the recording. So you don't need to worry about that because of network problem. But attendance, you must at least make an effort to join, because 3 times I will check your attendance, and I will only count those students who I find in this list. So we had talked, we had understood what the central dogma was. Central dogma of biology was that DNA is replicated as DNA. DNA is transcribed as nuclear RNA. The transcription takes place. Messenger RNA is formed, nuclear RNA that is processed, and messenger RNA is made. Messenger RNA is then translated, and the ribosome binds, and amino acid is made. This is the central dogma of molecular biology. So I would always suggest just download from Libgen from the site. We are all saying that we support Libgen Sci-Hub. Use Libgen Sci-Hub, download Gilbert's book. It's easily available over there. If you want, you can print out the particular chapter. That's perfect. That book is the only book you need to study for my part. Because the syllabus has been made looking at Gilbert. If you look at any textbook, you will not find where do I find these topics. You will not find these topics. You will find these topics. These are the syllabus. The people who made this this particular syllabus which you are studying, they opened the book of Gilbert, and chapter by chapter they put it in your syllabus and overloaded you. So basically, you can consider this as your textbook in developmental biology. So print the chapters which you have in your syllabus and read those chapters. You don't need any notes or anything for that. You can find it in Libgen. Libgen, type in Google Libgen and then open the Libgen site, libgen.rs. It may stop it because unfortunately there are a lot of publishing houses, corporate publishing houses. They are losing business because you are not buying their things. You are getting it for free. So they are basically they bribe or they convince or other ways, different methods. The government officials, they come and they tell them that I'll give you this much amount of tax. They are doing piracy. Please block their sites. And so they block the sites. Then you have to use some servers like Tor and all to go inside and access these. Or then you have to use proxies. So these are all corporate games to sell their products, aggressive marketing you can call. So all these things take place. Unfortunately, we are in no place to. It's not this particular any government anywhere in the world. This is how corporate companies work. They need to sell their products. Sad thing is that in a publication for which they are charging so much huge amount of money, they don't pay the authors, they don't pay the editors. So you are only just printing cost, and you are demanding such a huge amount of money unless you will not get information. So why is science is not reachable for everybody? Those who can only pay, they will only get science. So this is and for colleges like us, we will not get science. So therefore these sites like Libgen and all, they are very important. Of course, if they had kept their fees nominal, for example, maybe for this book they could have charged say 100 rupees. Everybody would have bought this online book for 100 rupees. No, they will charge 10,000 rupees for this. And why should I then buy this book? So if you charge it 100 rupees or 200 rupees, everybody would have bought this original book. So this is where the problem lies. Shuvarna has joined. Okay. So this is how so evidence actually of differential gene expression came with chromosomes known as Polytene chromosomes. So they are found in the salivary glands of certain larval tissues, Drosophila salivary glands or Chironomus salivary glands. In MSc you will find them. You will find that there are these Polytene chromosomes. These Polytene chromosomes have been found to have the same number of chromosomes and the same number of genes present in all the cells of the body of Drosophila. But the problem with Polytene means that the DNA divides. That the teen means the thread DNA thread divides, one becomes two, but they don't get separated into different cells. So result is, so this is one strand of DNA on top, beside there is no division, it doesn't separate, so it divides into two then divides into 4, it becomes thicker, then it starts dividing, it becomes thick, it becomes so thick, one chromosome of the salivary, it becomes so thick because of polytene, many threads of the same chromosome, it becomes so thick that you can easily see them under a microscope. You can stain them and you can find the banding patterns. So each band may be possibly one gene. It's the same in all the salivary gland chromosomes. That particular location of that particular band or that particular gene is the same in all the chromosomes, in all the cells. The same chromosomes, not all the chromosomes, in all the same chromosomes in all the cells. So this in Polytene chromosomes, this was perfect, but in different cells it was found in these polytenic chromosomes. It was found that there were certain regions where you can see in this picture, this is a Polytene chromosome band. There are certain banding patterns formed. Let me try to show you a Polytene chromosome. Let's open a window over here. A picture of a Polytene chromosome. Let's give a Google search and I will show you a Polytene chromosome. If you go to images, this is a Polytene chromosome. You can see different bands. These are arms, these are chromosomes in a Drosophila, for example. This is a giant Polytene chromosome. This one is a Polytene chromosome. So let's get back to the topic. So you see regions in the chromosomes, Polytene chromosomes, you will find regions of puffs. It's subtle, there is a puff comes out, dilutes. This was later found out by nucleic hybridization studies, where anti-complementary radioactive probe was given and hybridized on these chromosomes, and you could find that there were puff regions of particular genes. These were sites of active transcription of certain genes, and under different times of development and in different cells, these puffs were different. So this was a proof that of differential gene expression. Different genes are expressed at different times in different cells. So this was the first evidence of differential gene expression which has taken place. Even later studies in in particular, this was in Chironomus. In MSc, you isolate Polytene chromosomes in of Chironomus larva. You do that. There is a practical experiment which you do. Pankaj sir does it very well. Teaches all these practicals. So he does all these things. He will, if you study MSc in our college, then you will see, you will have the fortune of Pankaj sir showing you how to isolate. I have get these chironomous little red larvae. You have to take them, then on a slide under a binocular, you have to remove the head and pull out the salivary gland. Then you have to use a staining preparation of the salivary gland, and you have to squash, and then you see that polytene chromosomes, they spread. Some preparations are very good after a procession. Then you can see bands and patterns. You can see the polytene chromosomes. So in chironomous, but for me, in my practicals in BHU in MSc, we did Drosophila larva polytene chromosomes. But for here we use chironomous. More or less, it's this. Chironomous looks different. Drosophila is the one in the internet picture which I showed you. Lots of arms spreading about and all these things. So let us then we have this mRNA expression in Drosophila in stage 5 and stage 9. They are using two of the markers, 2 genes, one odd skipped related 1 gene. So odd skipped in stage 5. So they used a complementary probe for this mRNA. Opposite to the mRNA, complementary. So the complementary means they will bind to each other and one is labeled, and so you can detect. So you stain them. So you could see that odd skipped in stage 5, it's showing an expression. The deep blue expression is where this odd skipped expression is much higher. Then in stage 9, you see that pattern changes. You have much more number of cells expressed, but here it is concentrated in different regions. So these are evidences of differential gene expression which is taking place. You can see them in a particular embryo, in a mouse embryo also you can see that. Mouse embryo, you can see this blue region is this odd skipped, homologous of odd skipped in mouse. So this is how it goes. So let's go to the next part. You have heard about Dolly, nuclear equivalence. The final proof of genomic equivalence was demonstrated that the nuclei of differentiated somatic cells could generate any cell type in the body. So Dolly was a big example. So oocyte donor, the eggs were removed, and then you have the oocyte, empty oocyte, the nuclei was removed. See, the egg was removed from one of the this, and it was removed with a micropipette, and this was an empty egg. And from the udder cells, which is a somatic cell, they removed the nuclei and put it inside, and from this nuclei you got Dolly the sheep. Birth of Dolly. Dolly is dead now, but you got Dolly the sheep. So from that somatic cell which retained all the genomic complement, you could of an udder cell, it has already differentiated into an udder cell. From that you could get a new Kutti sheep here, Dolly. There were other problems which came with it, like arthritis and all, many things which came, the telomere shortening and things which were found, but you were able to get a clone a sheep from a different cell. This was a final proof of genomic equivalence, that all genes, all cells in your body have the same complement of genes, only they are differentially expressed, and therefore the different cells are different. This is how you express. So in the next part, I will start next day's class, I will start modulating access to genes, where I will be talking about nucleosome structures, histones, how genes are packed, and how genes are, what are the enzymes which are present. So terms like histone deacetylase, histone methylation, all these, remember, you study once, give 2 papers. It's there in eukaryotic gene expression as well as in differential gene expression in developmental biology. So study 1, get 2 results. So study this chapter properly, you study for 2 chapters. So this is how it goes. So with this I stop my class today. Let's see, I'll take an attendance once more. Who are the people who have attended and who have not attended? So Atika, Abir, Orpita, Oyon, Jahanara, Mollika, Mohor, Nazia, Papia, Paramita, Parthosarathi, Ria, Shalu, Sharoda, Sattaki, Shoma, Shreya, Subarna, Shubham, Shubham Shah, Ruidas, Shopnil. And of course, somehow I think Shantoni is having a problem with her internet today, but I will give her attendance. That's not a problem. It's the normal handset. The others don't attend class. So, any questions? Please feel free to ask about what I've taught today. Do you have any questions? Okay, if there is no questions, then I end my class over here. Thank you very much for attending.